So this video is the second one on your enzymes for CP biology. It's going to take you through part 3 and 4 of your enzyme guided notes. Please go back and make sure that you have everything filled in completed for parts 1 and 2. And if you do not, check with someone else, send me an email, but make sure we have those so you have a full set. So this one's going to go through how enzymes work and what can affect how well your enzymes work. So how do enzymes work? All right. So we already labeled in our graph this term substrate, and we just want to remind ourselves that substrate is what we call the molecule an enzyme is working on. So remember, in chemical reactions, we convert reactants into products. When an enzyme helps to break that reactant into a product, we rename the reactant. We rename the molecule because an enzyme is working on, and we call it a substrate. So if I wanted to use terminology to explain a chemical reaction where an enzyme is involved, I would say substrate converts into product. What's going to happen is we notice they've got this nice picture up here of our enzyme in bright pink. And our enzyme has this perfectly curved shape part of it. And this is called the active site. So the active site describes this perfect shaped surface on my enzyme. And if we look, my active site on my enzyme perfectly matches up to the texture and shape of my substrate, the molecule it works on. What's going to happen, and this is how all enzymes work, is the substrate is going to bind to that active site. So it's like they dock together or fit together perfectly, just like puzzle pieces. And they form something called the enzyme substrate complex. Now, when this is actually in place, so when the substrate is connected to the enzyme is actually going to help to break it down. So help kind of get it started turning into the product. And this is how enzymes actually achieve this lowering of activation energy. They basically, when they bind to a substrate, they basically already get that reaction started, get it going a little more quickly, so you don't have to add as much energy in order to convert it. So vocab term, we want to remember the surface where the substrate connects is the active site. And again, its shape is really important because it has to be perfectly shaped for the substrate. And when they're connected, they're the enzyme substrate complex. So I said this already, but it's just such a big idea, we have to reinforce it. That active site has to be perfectly shaped in order for the substrate to fit into it. If we alter the shape of the active site, the substrate doesn't fit anymore, the enzyme doesn't work. So substrate, and if we alter the shape of the protein, it's because of the substrate and enzyme not combining that enzyme ends up being ineffective, not being able to do its job very well. So we're in part 4 right now. Now we're going to talk about what are some things that can happen to an enzyme to actually cause it to change the shape of that active site. So the first thing that can happen, and this is an easy one, number 1 in our notes, if we change the DNA, so if you alter the code that was used to make that enzyme in the first place, well, then you make the wrong enzyme, it's the wrong shape, and it can't do its job. So absolutely, changing the DNA, change that in initial directions and how to make the enzyme can mess up your protein, mess up your enzyme, and make it not work perfectly. We're going to talk a little more in depth about pH and temperature, which are 2 other things that can affect the shape of our active site. So for pH and temperature, enzymes have an optimal, and you're going to see this word a lot. It's kind of a non-biology based vocab term for this, but we use it to describe the best temperature and pH that an enzyme works at. So we'll say, what is its optimal temperature? And we're just asking ourselves, okay, what's the best temperature this enzyme functions at? And same thing with pH. Outside of these optimal conditions, what can happen is your protein can actually denature or change shape. Denaturing is a vocab term that is used within enzymes to describe this process of an enzyme changing shape due to a change in pH or temperature. And if we kind of look at a picture of what that looks like. So here we are looking at an enzyme. So we notice that we're using color. So it's 3 different polypeptide chains that have come together to make this one enzyme. And here's my active site. So it's the surface right here. We notice it's got this perfect shape. The substrate would come in, it would have to bond to this perfect shape right here. Now, if I do something like add heat, or if I change the pH to make it really acidic or really basic, what happens is these strands all start to unravel. All of the bonds and connections that held it together in this perfect shape right here go away. And when it unravels or denatures, you'll notice that now I don't have an active site anymore. Now there's no perfect place for that substrate to fit in order to help that reaction happen more quickly. And this is why denaturing is dangerous and why it's bad, because you don't have an active site anymore for the substrate to bind to. We see another diagram down here. So your substrate is kind of this U shape and this V, and you have that perfect shape. And when then we apply heat, and look, heat changes the shape of that protein and its active site, because that's honestly the part of the protein we're most concerned about. We're concerned about that active site, that place where the substrate's going to dock. And now all of a sudden, nope, substrate can't connect anymore. Enzyme can't make the reaction happen faster. So what I would like you guys to do right now is check. You should have parts 3 and 4 all filled in. And in your notes, I want you to go through and see if you can interpret this graph. I'm going to check to see that we have filled in kind of all of these notes and at least attempted this. We're going to spend a lot of time talking about interpreting graphs, all that kind of stuff. But this one's on you. So email me if you have any questions and let me know